Subscribe my channel and press bell icon to get all notification.
Hello students, in this video we'll discuss about the next process related with bacteria, which is reproduction. So let's start reproduction in bacteria. When we talk about the reproduction in bacteria can reproduce by two ways. First, that is asexual reproduction and another is sexual reproduction. So we can find two types of reproduction in the case of bacteria. The first, which is asexual reproduction, and the another way that by which the bacteria can reproduce is a sexual reproduction. When we come on to the sexual reproduction, then we will find that there is not the actual sexual reproduction found in the bacteria. They are यहाँ पे ना तो gamete formation हो रहे हैं, they are not forming gametes, even no meiosis is taken place. So actual sexual reproduction is not found, but they reproduce somewhat like sexual snails. We will talk about it later. Now let's come on to the asexual reproduction. Asexual reproduction can be done by different ways. If we talk about this asexual reproduction, the first way by which the bacteria can reproduce asexually is binary fission. Binary fission. Binary fission is the most common type of asexual reproduction by which the bacteria can reproduce. सबसे ज्यादा बैक्टीरिया में जो रिप्रोडक्शन होता है वो होता है बाइनरी फिजन के थ्रू, एंड दिस टाइप ऑफ रिप्रोडक्शन इज डन ड्यूरिंग फेवरेबल कंडीशन. When the favorable conditions are present at that time the bacteria reproduce by the binary fission. Now let's see what happens in this process, what happens during the binary fission when a bacterial cell or a prokaryotic cell is reproduced by binary fission. This is a bacterial cell. We know in the bacteria the cell wall is present, cell membrane present, and inside the cell membrane cytoplasm, and inside the cytoplasm nucleoid is present. That is the genetic material of the bacterial cell. So nucleoid is present in the bacterial cell. At the time of division, when the time comes, the favorable conditions arise and the bacteria is required to reproduce. At that time, its nucleoid, that is the genetic material, this nucleoid get replicated. Nucleoid जो होता है उसके अंदर replication होता है. We know that in the bacterial cells a structure is found that is the infolds of the plasma membrane, that is known as mesosomes. So mesosomes present रहते हैं bacterial cell के अंदर, and the nucleoid remain attached with the mesosome during the time of reproduction. Reproduction के time पे, or we can say during the time of replication, it's remain attached with this mesosome. तो यहाँ पे हम देख सकते हैं कि ये mesosome है. This is the mesosome, and with this mesosome, the genetic material, the nucleoid is remain attached, and in this attached position, it replicates. It replicates and forms its own copy. ये replicate होगा and by replication it forms its own copies. तो यहाँ पे two copies form हो जाती हैं nucleoid के at the time of reproduction. After that, what happens? This mesosome gets divided. Mesosome get divided. In between this mesosome, a wall is get formed, or we can say the mesosome get divided. The mesosome two parts में divide हो जाएगा, and in this way we can say we will find such kind of position. This is the DNA and this is the another DNA or another nucleoid, and both of these two mesosomes are divided by this wall formation which happens between the mesosomes. So finally mesosome two parts में divide हो गया, and both of these two nucleoids which are formed, they remain attached with the mesosome. After that, that cell wall formation take place in the cytokinesis. Cell wall formation and cytokinesis take place in this bacterial cells, and we get two bacterial cells. Now we'll get two bacterial cells. Each bacterial cell which have its mesosome, दोनों ही bacterial cells में mesosome होंगे, and they will have their nucleoid. So in this way we can say that this is the way of reproduction where one bacterial cell is divided into the two daughter cells or two daughter bacterial cells. So this is the actual reproduction, the actual asexual reproduction by which the bacteria is dividing. So this is binary fission, and this is the reproduction which happens during favorable condition. If we talk about in the case of bacteria, it takes very less time to reproduce by binary fission. Hardly it takes 20 minutes time. Example, we take the example of E. coli, that is Escherichia coli, the bacteria. This E. coli bacteria can reproduce by binary fission within the or it completes its binary fission within 20 minutes. 20 मिनट के अंदर binary fission complete हो जाता है. Out of these 20 minutes, the 18 minutes time takes to replicate the nucleus, and after that the 2 minutes time is taken just to divide these bacterial cell into the two daughter cells. Overall 20 minutes के अंदर एक bacterial cell divide होकर two daughter cells form करती हैं, and after that these daughter cells will again divide within next 20 minutes. So just within 20 minutes, they can reproduce by binary fission. So this is the one way of asexual reproduction that is found in the bacterial cell. The another way of asexual reproduction is spore formation or sporulation. Second way of asexual reproduction, second type of asexual reproduction we can say is a spore formation. The another way of asexual reproduction is sporulation. So we will discuss the sporulation. Sporulation is the method by which the bacteria can reproduce asexually during unfavorable condition. Like binary fission is the reproduction which happens during favorable condition, when the food, temperature, all things are favorable. But during unfavorable conditions, bacteria reproduce by sporulation. They can form different types of spores like conidia, cysts, endoscopes, etc. तो ये जो spores formation होता है, they happen during unfavorable conditions, when the food is not available, or we can say the temperature is not suitable for the bacteria, or the toxic substances are present in the surrounding. At that time, the bacteria form spores. So we'll discuss the asexual reproduction by endoscopes. Endoscope, that is a type of spores formed mostly in the bacteria, by which the bacteria reproduce asexually. Let's see what happens during endoscope formation. During unfavorable condition, the bacterial cell, inside the bacterial cells is nucleoid that is present into the cytoplasm. This nucleoid comes or reach to one side of the bacterial cell. एक side में इकट्ठा हो जाता है nucleoid, and then with some dense cytoplasm, it's kept some dense cytoplasm and make a cover and get covered by a wall. एक wall formation हो जाती है. Here, what happened? What get collected here? The nucleoid with some dense cytoplasm. It's released. It get dehydrated. Water release कर देगा, dehydrated हो जाएगा, and then cytoplasm यहाँ पे nucleoid के साथ collect होगा. Around this dense cytoplasm and nucleoid, a wall is get formed. This wall is a wall which is very resistant wall. It gives protection to this nucleoid. By this way, it formed a structure. That structure is known as endoscope. इस structure को हम नाम दे रहे हैं endoscope. यहाँ पे that the remaining bacterial cells, this remaining bacterial cells, the cytoplasm is present. But when the condition become more adverse, बहुत ज़्यादा critical condition जब आ जाएगी, at that time these cells get died. Cytoplasm भी सारा इसका rupture हो जाएगा, and it will get died. Cell gets died, and this endosperm is released. Endosperm release हो जाएगा, and this endosperm can tolerate all the adverse condition. जब भी adverse condition होगी, चाहे वो किसी भी तरीके की हो, temperature हो, toxic substances हो, it can tolerate because it has a layer or layers of the wall which are present around it. यहाँ पे wall present होती है, अंदर nucleoid होती है, and then this part of the cytoplasm which is present here is the cortex part. So ये सब contain किया हुआ endosperm है, endospore है. This endospore can tolerate the adverse condition, and when the condition become favorable, at that time it will break these walls and this nucleoid come outside. It will form this nucleoid come outside, and then this bacteria will again during the favorable condition will divide by binary fission. So here you can see that this is the type of reproduction, but actually here not any reproduction is happening. यहाँ पे कोई reproduction नहीं हो रहा. This endosperm is just a structure which provide protection. सिर्फ protection provide करने के लिए बनी हुई एक structure है. It is providing protection against adverse condition, against unfavorable condition. But when this endospores overcome by this unfavorable condition, then it can reproduce. It will reproduce and it form the new bacterial daughter cells. तो इसलिए हमें बोल सकते हैं कि है reproduction. इसके बाद reproduction हो रहा है, but it's not actually the part of the reproduction. It's not a structure that is responsible for the reproduction. Such type of endospores are found in some pathogenic bacteria. कुछ बहुत critical कुछ pathogenic bacteria होते हैं like Clostridium tetani. which causes the tetanus disease. Clostridium tetani is the example of the bacteria which form such type of endospore, and these spores are not get affected by any toxic substances. किसी भी toxic substance का इनके ऊपर effect नहीं होता. So this is the way by which we can see that bacteria are dividing by asexual means. We discussed two methods. First, that is binary fission, and second by endoscope formation. Now let's come on to the next type of reproduction which is found in the bacteria, which is sexual reproduction. So now we'll discuss about the sexual reproduction. Now let's see sexual reproduction in bacteria. Sexual reproduction. When we talk about the sexual reproduction in bacteria, there no gametes are forming, no meiosis take place. किसी भी तरीके का वो चीजें नहीं दिखाई दे रही जो actual sexual reproduction में होता है. So we can say here the true sexual reproduction is not found into the bacterial cells, but genetic recombination happens. यहाँ पे genetic transfer होगा, genetic DNA we can say, or genetic materials get transformed. That's why we are calling it sexual reproduction. So here you have to keep remember that this is not actual sexual reproduction. Here just transfer of genetic material happens. That's why we are calling it sexual reproduction, or we can say parasexual reproduction. So this is the reproduction which happens into the bacteria. Bacteria can reproduce by this way by three methods. तीन तरीके हैं जिनके थ्रू बैक्टीरिया सेक्सुअली रिप्रोड्यूस कर सकता है, और द ट्रांसफर ऑफ जेनेटिक मटेरियल्स हैपेंस by the three ways. The first way is the transformation. Transformation. This method is explained by the Griffith. The name of the scientist who explained this method is the Griffith. Griffith explained this method of reproduction into the bacteria transformation. In the transformation, the bacterial cells take the genetic material or the DNA from its surrounding. So this is the way, or this is the method of reproduction where the genetic materials come inside or taken by the bacterial cells from its surrounding. Griffith ने इसके ऊपर बहुत एक्सपेरिमेंट करके प्रूफ किया था by this transformation. Griffith taken many experiment and he proved that DNA is a genetic material. इससे पहले ये नहीं माना जाता था कि DNA genetic material है, but Griffith proved that DNA is a genetic material through this transformation experiment. So transformation is a process in which we can say that DNA is taken inside or taken by the bacterial cells from its surrounding. So here the transformation happens. This is explained by the Griffith, and the proof, the biochemical proof of this transformation was given by the scientist. That name was Avery McCarthy. Avery McCarthy and McLeod. These are the name of the scientists who explained that this transformation, that McLeod, who explained or who given the biochemical proof related with this transformation. So this is the way by which we can say that bacteria is taking genetic material. Second way of sexual method is the conjugation. Conjugation. Now, what happened into the conjugation? Conjugation is the asexual reproduction or the transfer of genetic material from one bacterial cell to another bacterial cell. Let's see what happens during this conjugation process. First, let's say this process is discovered by or explained by Lederberg and Tatum. The name of the scientist who explained this process was Lederberg and Tatum, and we explained this process into the E. coli bacteria. Now let's see what happens during conjugation process. Into the conjugation, one bacterial cell. This is one bacterial cell, and this one is another bacterial cell. These two bacterial cells, they come closer and they form a bridge-like structure. This bridge is known as conjugation bridge. They will form a bridge which is known as conjugation bridge. यहाँ पे conjugation bridge बनेगा. At that time, here the two bacterial cells which are coming together, one is the positive strain and another one will be negative strain. यहाँ पे positive और negative strain जो हमने लगाया है, that shows their fertility. The presence of fertility gene on their plasmid. Bacteria के अंदर nucleoid present होता है. It has its nucleoid. Besides nucleoid, they have some extra chromosomal DNA. That extra chromosomal circular DNA, which is known as plasmid. Plasmid present होगा. And on this plasmid के ऊपर, this is the plasmid, and this plasmid contains some specific genes which are known as F gene or fertility gene. So the presence of these F gene onto the plasmid makes the bacterial cell positive. उसे हमने शो किया है पॉजिटिव और we can say that this is F positive because the fertility gene is found in this bacteria. So we call it F positive. When this F positive bacteria come closer and make a conjugation bridge with another bacteria which is F negative, जहाँ पे fertility gene present नहीं है, इस टाइम पे क्या होगा? At that time, this bacteria will transfer its genetic material from one bacteria to another. इस टाइम पे एक बैक्टीरिया से जेनेटिक मटेरियल ट्रांसफर होके दूसरे बैक्टीरिया के अंदर जाएगा, and the transfer of genetic material which happens through this conjugation bridge. In this way, the two-way can be adopted for the transformation of genetic material from one bacteria to another bacteria. One way is that in which the genetic material get replicated. It's make two copies. One copy is transferred to this bacteria. एक कॉपी यहाँ पे जेनेटिक मटेरियल है, इसकी एक कॉपी बनी and this copy is transferred to this another bacteria, that is F negative bacteria, जो कि fertility genes में present नहीं थे. In this way we can say that both of these two type bacteria will become the positive strain. दोनों ही bacteria positive bacteria बन जाएंगे. Another way, second way of the transformation or another way of the transfer of this genetic material can be that genetic material is transferred as it is, जिसमें replication नहीं हो रहा. In that way, what will happen? This type of bacteria, this another bacteria, this will become the positive and this will become negative. तो उस केस में एक positive बन जाएगा और एक negative बन जाएगा. So this is the way of reproduction by which we can say that genetic material is transferred from one bacteria to another bacteria. अब अगर हम इसे reproduction से related करें, so here we can say that there are no total cells are forming. कोई भी total cell नहीं बनी, तो हम इसे कैसे reproduction कह सकते हैं? Here reproduction is happen when this bacteria, this F negative bacteria, when it get the fertility gene. जब इसे fertility gene मिल जाते हैं, when it get the fertility gene, after that it will divide by the binary fission and it will form the daughter cell. So in this way it is related with the reproduction. And this F gene is responsible for the reproduction because due to this presence of this F gene, they got the power of fertility. They can form this pili-like structure by which they can come closer with the another bacteria and then they can form a conjugation bridge. So this is the second way of the sexual reproduction, which is conjugation, explained by Letterberg and Charter. And here the fertility gene should be present in one bacteria through which the genetic material gets transferred to the next bacteria or another bacteria which is F negative, which is lacking the fertility gene. So this is the two way. The third way of sexual reproduction is the transduction. Transduction. Transduction is the way or is another way of the transfer of genetic material where the genetic material gets transferred by the help of viruses. And the viruses which are used here are known as bacteriophages. Bacteriophage. Bacteriophages are the viruses which depends onto the bacteria. So that's why we are calling it bacteriophage. And these bacteriophages are responsible for the transfer of genetic material from one bacteria to another bacteria. एक host bacteria से genetic material को carry करेंगे and then these bacteriophages will transfer this genetic material to another bacteria. And this process is explained. This transduction was explained by Ziller and Letterberg. The name of the scientists who explained this process is Ziller and Letterberg. So these are the name of the scientists who explain the process of transduction, where the transfer of genetic material happens through the viruses that are called as bacteriophages. So here we discuss the method of reproduction into the bacteria. Bacteria can reproduce by asexual and the sexual way. In the asexual, the binary fission is the most commonly method by which the bacteria reproduce. But when we talk about the sexual reproduction, the two type of sexual reproduction is not found. This type of reproduction which is found is also known as parasexual method. So we can say that parasexuality is present into the bacteria, where the genetic materials gets transformed from one bacteria to another bacteria, but actual sexual reproduction do not take place. Here three markers are present, where the first marker of the sexual reproduction, that is transformation, where that genetic materials is taken from the surrounding. Second marker is the conjugation, where the genetic materials transport from one bacteria to the another bacteria through passing through the conjugation bridge. And the third method of sexual reproduction is the transduction, where genetic material is transferred through the viruses or with the help of viruses which are known as bacteriophages. So this is the reproduction into the bacteria.